So yeah, hi everyone. Today we're joined by Dr. Johanne from the University of Edinburgh, and she's going to be talking to us about some work from the iAtlantic research project. So without any further delay, take it away, Johanne.
Hi everyone, thanks Catherine. Thank you for joining me for this talk, and thanks to JNCC for giving me the opportunity to present here today. So as just mentioned, I'm Johanne. I'm a university teacher at the School of Geosciences here at the University of Edinburgh, and I will present to you today some results from the iAtlantic project, which was my previous postdoc. Before I go into these results, I just thought I would give you a bit of background about myself. So I'm a benthic ecologist by trade. I worked on cold water corals and sponge grounds during my master's and PhD studies, and I mainly used traditional survey data, so image and video-based data, combined with numerical ecology analysis method to try and study, I suppose, these really complex habitats. So looked at how we can try and establish what healthy colonies look like in cold water coral reefs. I also worked quite a bit on the Faroe Shetland Channel to try and understand what the impact of both bottom trawling, but also oil and gas infrastructures in the area have on the sponge grounds. And then more recently, I've continued this work with collaborators looking at the use of time-lapse photography to determine the impacts of oil and gas activities on deep sea sponges. In addition to this approach, or these approaches, I've also looked at how we can use molecular data to improve our understanding of these benthic organisms. So I've run experiments in the lab where I've exposed sponges to oil and try and understand both the gene expression response of the sponge host through these kind of heat maps. I was just saying that, yeah, using these kind of RNA sequencing techniques, you can try and figure out the gene response of the sponge host and also the microbiome that lives inside the sponge. And I'm really interested in figuring out how we can use some of these techniques in the field. So not just in very controlled experiments, but also using them in nature to try and monitor these deep sea ecosystems. I guess the thing that really brings me to bring these two approaches together in my mind is data analysis. So I really enjoy making sense of data, and this is what brought me to the iAtlantic research project. So iAtlantic is a Horizon 20 research project that's ending in about 6 months now. It's quite a big project. It had it has 35 partners and 12 associated partners. You can see some of the logos here, and they're really distributed across the whole of the Atlantic basin. The project had 12 study regions, which are kind of highlighted in yellow here on the map. So you can see really trying to cover a breadth of the Atlantic Ocean. The project had 5 different objectives. And today I'm really focusing on objective 3, which was to conduct an ecosystem assessment of open water and deep sea ecosystems over space and time. I worked on the temporal aspect of the project. And so the first step really for us was to compile all of the ecological time series data that were available throughout our study regions. So they go from plankton time series data all the way up to whales. They had really varying degrees of resolution in terms of how often samples were taken, but also duration, so how long the time series was collected for. And so it was a really kind of wide mix of data that we had available for the project across all of these different time study regions that we were trying to make sense of. We had 3 main overarching objectives across all of these different data sets. So it was to identify any temporal trends, identify some breakpoints, and then finally relate these to changes in the environment. So this work package was led by Dr. Leanne Henry from the University of Edinburgh and Marjolaine Matabeau from Ifremer. And we were guided by Professor Pierre Legendre, who is one of the founder of numerical ecology, and it was really great to have his advice for making sense of all of our data. You can probably guess from this picture that all of this happened during COVID. So In one way, that was a blessing that we were working on data sets that were already gathered. In another way, it did make our work quite a bit more complicated, but managed pretty well to have a common approach across all of our data sets. And this common approach is summarized in this kind of decision tree that we came up with. So these kind of decision tree allows you to select which analysis is needed to be run depending on your, first of all, research question, but also the type of data that you have available. So these decision trees are currently available as deliverables for the project, but we're hoping to get a paper out soon to try and explain how statistics can be used to make sense of time series data. So more focusing on my work, I was responsible to analyze data from two study regions, the Scotian Slope here offshore Nova Scotia, and then the Sargasso Sea. From the Scotian Slope, we have zooplankton and demersal fish and invertebrate time series. And you might have been wondering why I had a lovely picture of a squid on the invitation for this talk. And I've tricked you today on Halloween. This talk isn't mostly statistical, but I needed a cool picture. And one of the species that we follow from the Scotian Slope is ilex. So I picked this one from a collaborator in DFO, from which that gave us the data. But I must admit, it's the only cool picture I have of an animal on my slides. So apologies for that, but hopefully you'll still find it interesting. The second study region, the Sargasso Sea, we also had access to phytoplankton, zooplankton, and carbon transport data. In the next few minutes, I'm just going to go through some of our main results and give you more details into the data sets for each of these study regions. So starting with the Scotian Slope first. So this study region was defined as the 200-meter depth contour to the limit of the Canadian EZ. It's in red here on the map. And it includes the Gully Canyon, which is a really amazing submarine canyon. It's the largest submarine canyon in the North Atlantic. So it's really prominent feature on the bathymetry of the region. The other thing to notice for this region is that it's also adjacent to the Laurentian Channel that you can see here again on the bathymetry. And this means that the region is characterized by really complex oceanography. So you have water derived from the Gulf Stream coming from the south of the region, mixing with currents derived from the Labrador Current, and as well as water coming out from the Laurentian Channel. This complex oceanography supports really high levels of biodiversity. So the study region is on migratory routes of pelagic fish and encompasses overwintering grounds for demersal fish. It also holds significant population of corals and sponges on the canyon and the slopes. And finally, the canyon itself, which is a marine protected area since 2004, is known for its diverse population of cetaceans. Overall, the region has seen quite intense fishing on the continental shelf and the upper slope between the 60s and early 90s, but activities have since declined, mainly due to a decline in stock. Before I carry on into the results of for this study region, I do want to highlight the contribution of my co-authors from DFO and the Spanish Institute of Oceanography. So what data set did we actually have access to here? So in terms of environmental condition, we looked at remotely sensed sea surface temperature and chlorophyll concentrations. We also had access to some modeled outputs, but I'm not going to cover this in my talk today for time constraints. We also had access to zooplankton, which were derived from a monitoring survey program called ASUMP, and this gave us abundances for 378 taxa in spring and fall between 1999 and 2019. So quite a lot of data here. I also had access to some demersal fish and invertebrate surveys. So these were random stratified trawl surveys that were deployed yearly between 82 and 2018 and provided us with abundances for 96 taxa. So going through each of these data sets, what were the main results? Well, first, if we look at the remotely sensed sea surface temperature for fall here in orange, spring in green, and annual in black, we see that there's a really high level of interannual variability, which is expected for such a oceanographically complex region. We also noticed that there is a significant increase in the annual sea surface temperature average over time. That's denoted by this regression line. If we try and simplify a little bit our time series and we compare the beginning and the end of the time series, we do find some significant differences for annual and fall sea surface temperature between the first and the last 5 years. We don't find this difference due to in the spring because of the high level of noise in our time series. But overall, we do see an increase in temperature, which we were expecting based on literature for the region. What about the zooplankton? Well, the first thing to say is that the zooplankton biomass in the region is really much dominated by copepod species, so mainly Calanus finmarchicus, Metridia lucens, and Metridia longa. And this is the case for spring and fall, with obviously some differences between those two seasons, which we would expect. But the fact that these zooplankton are dominating the community, even at the beginning as well as at the end of our time series, don't mean that there's no changes in the rest of the community. So if we look at the abundance, we again see a really high level of interannual variability. Here, these plots are outputs for what's called a community trajectory analysis. And they might seem really complex and messy, but they're actually really easy to read. So each point on this trajectory represent the status of the community through time, starting in 1999 and kind of continuing through the early 2000s and then all the way over to 2019. And that's the same for the fall. So starting in 99 and moving all the way across to 2018. And we can see that there's large jumps between a lot of these years, meaning there's quite big differences in the community composition between those two time points. And these jumps might not reflect any kind of long-term trend in change, but simply that the zooplankton community is highly variable from one year to the next. Using a separate analysis called multivariate regression trees, we also identified breakpoints in our data sets. And that was in 2013 in spring and in 2008 in fall. And you can actually see these breakpoints appearing on your trajectory, especially in fall, where you see that after the breakpoint, the trajectory starts having a completely different behavior to before 2008. And the thing to notice really on these trajectories is that despite the high level of noise, you still have quite a directional trajectory going from one side of your plot to the other side. And this is especially strong for the fall data set. So we can see significant changes in the zooplankton community over time, despite the level of noise. Okay, so we know that there's changes, but what are those changes actually, I guess, doing? So to try and investigate this, we looked at, we applied another analysis called dynamic factor analysis, which tries to identify common trends across different taxa over time. This dynamic factor analysis picked up one single common trend in spring and fall. And here the y-axis is standardized abundances. So when this curve goes below zero, it means that you have below average abundances. This happens in 2010 for spring, but with this really strong decrease in 2014, our breakpoint. And it happened, let me just move this little window here, and it happened in 2008 for fall, again, our significant breakpoint that we had already identified. Each taxa modeled through this dynamic factor analysis can either follow this exact trend or the opposite of the trend. And so we found that we could model 10 taxa increasing over time in spring and 9 decreasing. And we could model 8 taxa increasing over time in fall and 23 decreasing over time following this exact trend. So we have the kind of groupings of species that are increasing and decreasing over time, but what does that actually mean for the community? And so we tried to look a little bit at some zooplankton traits to make sense of these increases and decreases. What we did was we calculated the community temperature index, which basically reflects if you have more warm water or cold water species in your mix. And what we see is that when we regress this for the spring data over time, with sea surface temperature, you had a really clear increase in the community temperature index with temperature of the water, meaning that you have an increase in the prevalence of warm water species. We also calculated the overall carbon content of our community. And here we see the opposite. We have a decrease, a quite strict decrease in carbon content with increase in sea surface temperature. Again, for the spring data, the fall isn't really showing us a similar trend. And this means that as the temperature has increased in our study region, we see an increase in warm water species that are poor in carbon overall. Okay, so this is the changes that we've seen for the zooplankton in our region. Let's quickly go over our fish. So if you thought that our zooplankton trajectory was messy, you've not seen anything. The fish is even worse. So this is really where we see a really high level of noise. So we're moving from 1982 on one side of the trajectory across to 2018 on the other. And you can see these really big jumps from one year to the next. We did find 2 breakpoints through multivariate regression trees, so in 96 and in 2012. But I would really challenge you to actually pick these out on the trajectory. Because of the high level of noise, it'd be really difficult. And what I will say, though, on this trajectory is despite the fact that it jumps back and forth, it does overall move from one corner of the plot to the other side of the plot. So we are seeing some significant change, but really difficult to make sense of due to this level of noise. If we look at our dynamic factor analysis, so again, an analysis that tries to find common trend across all of our species, we actually find 2 trends here, really highlighting the very complex behavior of our fish population in this region. The first trend is increasing over time until 2012, one of our breakpoints. The second one is overall decreasing, but with this really strange behavior between 95 and 2000, again, at the time where we picked up a breakpoint. So we have good agreements across our statistical analysis, which is always reassuring to see. Again, we can then look at which species follow what trend and in what direction, and try and form groups of species increasing over time or decreasing over time. And what we see is when we look at their thermal preferences, yes, groups that are increasing in abundance seems to have higher thermal preferences than groups decreasing in abundance, but it's really noisy, there's lots of variability, and we need to look at other traits to try and make sense of this data better. So we still have some work to do on the fish data. So I hope I've shown you some interesting trends in our Scotian Slope study region. I'd like to just show you a few results from the Sargasso Sea, mainly showing some different analysis that you can run with data that are sampled not just seasonally or yearly, like in the Scotian Slope, but every month, which is a really amazing type of data. So for the Sargasso Sea, we actually use data from the Bermuda Atlantic Time Series, or BATS station, and it's located about 75 kilometers to the southeast of Bermuda. The station has seen monthly cruises since 1988, which has measured a range of hydrographic and biological variables. And the sampling happens throughout the water column, and the really great thing is that all of this data is open access. So any one of you could go onto the BATS website, download the data and analyze it. The main objective for the BATS station is really to improve our understanding of biochemical cycling of carbon in the North Atlantic Ocean. You might have seen some of the BATS data already plotted up, and it's often used to illustrate changes in environmental conditions due to climate change. So this figure is from a paper that was published in 2020, showing you the increase in sea surface temperature and salinity, as well as the decrease in dissolved oxygen concentrations, increase in dissolved inorganic carbon, and anthropogenic carbon. So this data is quite well known, but we've really focused on the ecological part of the time series. And here again, I just want to highlight the contribution of my collaborators, Nick Bates from BIOS and Leanne Henry from the University of Edinburgh. So let's look over at the data that we analyzed. So we looked at CTD casts collected between 1989 and 2016, where we got monthly measurements of temperature, salinity, dissolved oxygen, etc. We also analyzed some phytoplankton data. So I derived some abundances from pigment concentrations, and these were available between 1995 and 2019, and that gave us a proxy for abundance for 7 taxonomical groups. And finally, zooplankton. We had access to direct enumeration for 5 different size classes, so that gave us monthly biomass between 94 and 2020. So you can see how each of our dataset doesn't really fully fit the same temporal resolution, but there is a good chunk of years that are common between them, which allows us to kind of compare and contrast what the changes that have happened in each of these datasets. I want to show you some main results and hopefully give you an idea of what other types of analysis you can run with temporal data. So I did look at shifts in community composition the same way that I did for the Scotian Slope. And we did see an increase in the prevalence of small phytoplankton groups that we could directly link to changes in temperature, dissolved oxygen, and CO2 concentrations. But today I really want to focus on changes in the phenology of these time series. So I looked at the timing of the bloom peak in spring and fall for different compartments of the plankton groups. So for the small phytoplanktons, which are increasing in prevalence, what we see is that over time, the spring bloom peak is actually being delayed, whereas the fall bloom peak is coming in early. So you have a reduction in the lag between the 2 annual blooms for this compartment. If we look at the large phytoplankton groups, we see again that the spring bloom peak is delayed with the years, but the fall bloom peak is also delayed. So here we see more of a shift in the timing of the annual blooms. If I now look at the zooplankton, again, we see some changes in the community composition with the prevalence of larger zooplankton groups becoming more abundant over time. And again, we can link these with temperature, changes in temperature and salinity, as well as CO2 concentration. But if we look at the phenological analysis, we see that there's really no shift in the timing of the zooplankton blooms, both in spring and fall. So what does that leave us with? It kind of leaves us with a bit of a weird system where the within year variance between the different blooms is increasing over time, especially in the fall data set, but also significantly in the spring. And so it does hint at a bit of a mismatch being created between your smaller phytoplankton, your larger phytoplankton, and your zooplankton. Be really interested to dig further into this. And one way we can maybe look at it is also to see how this impacts carbon transport to deeper settings. I'm obviously particularly interested in this as a benthic ecologist. You do wonder what these changes in the upper reaches of the water column will have as consequences for the benthic communities. Very briefly, what we see when we look at the amount of organic carbon that gets transported to 150 meters depth over time, and this is data coming from sediment traps, we do see overall an increase in the amount of carbon that is provided to the benthic community. And this might be maybe driven by the increase in the size of the zooplankton groups. Be really interested to dig further into the carbon to nitrogen partition of this material. And this is data that we do have access to, but I've not had the chance to fully look into yet. Very briefly, I just want to highlight that this two analysis, or the two study region that I showed results for today, is really not all that's been done within this work package. We have a whole host of publications that have come out in recent years from the iAtlantic research project. I think I'd particularly like to highlight the work from our Brazilian partners who have used fisheries landing data to show how you have tropicalization of the demersal megafauna in the South Atlantic since 2013. So this is a really interesting paper showing how we can use unconventional data sets to look for temporal signals of change. So it's a really great effort from the Brazilian partners. In conclusion, this has been a bit of a whirlwind tour of my 2 iAtlantic study region, but I hope you've seen that we've found really significant shifts in both study region, and these have mainly been driven by changes in temperature. And these shifts are not just shifts in community composition, but also in species traits, so their size, the temperature affinities, as well as their carbon content. And we've also seen that there's changes in the timing of phenological events. I also hope that I've showed you that you can use really varied statistical analysis to interrogate noisy, sometimes partial time series data. And this is particularly important for open water and deep sea studies where we might not have the best resolution always available to us. But with clever statistics, we can really make a lot of sense of these data sets. For me, the next steps are to adapt the approaches I've shown you today, such as the trajectory analysis, to the omics dataset, so the RNA sequencing and other sequencing datasets. So that's kind of what I've shown you in the beginning of my talk, kind of gone full circle. And this is really to allow their use in field studies. So this is something that's of great interest to me at the moment. And I'm preparing fellowship applications to try and do this, one of which is due tomorrow, which is a bit stressful. But if you are interested in these kind of themes, please do get in touch, because I'd be very interested in discussing this further. And that's all from me. I hope that has reached you reasonably clearly without any more internet interruptions.
Thank you so much, Johanne. That was really interesting. And yeah, good luck on the fellowship application as well. So we have a few questions from people that are posted on the Jamboard. So I was wondering I could just go through them, and yeah, feel free to answer them as you'd like. So the first one is, what are some of the likely causes of such big changes in zooplankton communities over 1 year?
So it might not actually be that we're seeing any changes. It's probably down to our sampling. So especially if we think of the fall, but it could be true also in the, especially in the spring, but it could also be true in the fall. The ship, if you will go out every year around mid-April. But the timing of the bloom, especially, well, in both study region, but it's particularly true in the Scotian Slope, the timing of the bloom will switch a lot from year on year. And so 1 year you might hit the bloom perfectly and you get a sample that represents the spring bloom really well. But 1 year you might be a bit too early and you might have a sample that represents more what's happening in the winter months, or vice versa. You might hit a post-bloom situation where you might have something that represents later spring, summer communities. So these big jumps in the data isn't necessarily showing ecological variability, but really just noise and the fact that we're not out there all the time collecting samples so often. And that's where these monthly samples can help us figure out, are we hitting the bloom yet, or are we post-bloom or pre-bloom? So that's probably the reason we're seeing these big jumps.
Okay. That makes sense. Thank you. And how were the plankton samples collected?
So for the Scotian Slope, it's vertical ring nets from kind of, I can't remember the depth from top of my head. For the bats data set, so it's 2 different methods depending on the phytoplankton and the zooplankton. So phytoplankton, it's based on, it's from the CTD rosettes in both situations. For the phytoplankton, it's really pigment concentrations. So there's a whole range of pigments measured in our water samples collected through the CTD, and then we can manipulate those to try and get abundance proxies for different large groups of phytoplankton. For the zooplankton, it's direct enumeration. So these get picked out of the water samples and then measured in terms of biomass, depending on their size. So it's quite a big of an easier process for that one. But we don't have taxonomical information, which is a bit of a shame.
Okay, I see. And what future research will happen with the Scotian Slope fish data?
Yeah, I mean, the surveys are carrying on. So this is data that that DFO is collecting actively. There was a bit of a break because of COVID. It was really difficult to get the ship out. There was also some issues with the research vessel, but the surveys are continuing. So we will try and make sense of these really noisy data set that we have and those strange dynamic factor analysis trends that we have found. And then hopefully as the surveys carry on, we'll be able to make more and more sense of these shifts. This is really the danger of temporal analysis, is you always want the next time point, and you feel like that will explain the overall trend that you're seeing. I think that's like a forever quest to get one more time in the analysis and see if that changes everything. So yeah, the data will still be gathered and analyzed by DFO partners in the future.
Okay, sweet. And why is there so much noise in the time series data? That's another question.
Oh yeah, it's something that we've discussed a lot across the whole of the project, and I have colleagues here at the University of Edinburgh that are not necessarily part of iAtlantic, but they also look at time series, and noise is kind of the pain of our existence, really, when you look at these data sets. Part of it is the fact that part of it is that we're looking at very complex systems. So especially if we think about zooplankton, the reason why they are often used as proxies for what changed in the water column is because they react so quickly to a shift in temperature or another water mass entering the system transports different species of zooplankton. So first reason is the systems are just incredibly dynamic, and so there is a lot of variability naturally there. The second one is kind of going back to I think the first question that was asked is our sampling. So depending on when we sample in relations to bloom events or other biological events, we might get a slightly biased vision of the community that year. And so that can kind of look like variability, but actually it's just the fact that we've not quite managed to sample quite the same point over time. So that will increase our variability as well. I think that's the two main reasons that I can just think of. I'm sure there's plenty more. There's probably some improvement to equipment as well that we're using that can help us get better data sets. So I know with the sediment traps, for example, that there's a bit of uncertainty about how good they are at measuring what's falling down from above. So yeah, I think that's probably the three main reasons. Natural variability, just timing of biological events, and then equipment.
Okay, great. So we've had a few more questions pop up, which is good. So how did you define spring and fall? Do you think selecting an arbitrary month is useful, or would it be better to define it by the environmental abiotics?
Yeah, that's a really good question. So we ranged it over 3 months to try and capture a little bit this variability in the sampling time, etc. You could try, as you said, to look at what's happening in the environmental data and decide when spring has hit that year, if you will. The risk there is that you might actually almost bias your analysis because you've kind of selected for what you want to see. So then when you relate back to the environmental variables, you've already kind of selected the data points that works best for what you. So there's a bit of a trick there between biasing too much your analysis versus trying to make sense and being more representative of what's happening. So we've used a very, very kind of blunt cutoff in terms of our spring running from, I think, April, May, June, and looked at it that way. But yes, that could be another approach to look at this data.
Okay, I see. And did you consider shortening the study time period for the different taxa so that the time series were directly comparable, or what was the argument for leaving them as is?
Yeah, we've done kind of both. So we've tried to maximize the amount of time points that we had for especially for the ecological data sets, and then we've kind of cut the bits where we had continuous or similar coverage across. So looking at individual data sets, we've tried to maximize the amount of time points, gives us more information. And then when we've compared across, we've kind of looked at just what we have in common, which we have to. So kind of two-pronged approach. It really depends on your, I guess, the research question, but we've decided to do both. Yeah.
Okay. Great, thank you. And I think the last question, can you use data for predictive modeling of what future zooplankton/fish populations may look like? And is this going to be done soon, if so?
Yeah, so the dynamic factor analysis allows you to include environmental variables into the model, and you could then project in the future. And this is just my own interpretation of the analysis. I think some of my co-author might not agree on this one. I am a little bit worried about projecting because I feel like we don't have a full understanding of the changes we're already seeing. There's also some uncertainty around the environment, how the environmental variables are going to change as well. So when we use that to project our ecological data, it becomes really complex, and the uncertainty increases a lot. So it's not something I'm going to run personally. It might be done in the future. I mean, there are already some work that have tried to predict what changes will occur, especially in the plankton, which does seem to fit with some of the change we've seen here. So the decrease towards smaller phytoplankton has been described in the literature through modeling work, and the predicted increase in more gelatinous plankton as well. And that's what we see when we look at the carbon content of our community. So what we're seeing so far does seem to fit with projections. Yes, the data could be used for projection, but I am a little bit careful about it. But maybe that shows my lack of, it's not something I typically do. So maybe that's what that shows.
Okay, that's fair enough. Okay, so I think that's all the questions. If anyone kind of last call for questions posting on the Jamboard. I think that's all of them now. But yeah, thank you so much, Johanne. That was really interesting.
Thank you. No, thanks for joining. No, I appreciate it.
Thank you, Johanne. Thanks.
Thanks, Catherine.